to the around the world. Frontline of AAV vectors for gene therapy research in Japan. Today's webinar will explore cutting-edge advancements in the use of AAV vectors for gene therapy research in Japan. This event brings together leading scientists, researchers, and professionals in the field to discuss innovative approaches, challenges, and breakthroughs in gene delivery systems, specifically focusing on AAV technology. All questions will be saved until the end when both speakers will answer questions asked throughout the presentations. You are encouraged to submit your questions throughout the presentations using the Q&A tab located to the right of the video player. You may continue to submit questions throughout the Q&A discussion at the end of the talks as well. We have two amazing presenters today. Our first speaker is Dr. Hirokazu Hirai. He earned his MD from Kobe University in 1989 and completed his PhD there in 1994. He conducted postdoctoral research on NMDA type glutamate receptor structure at the Max Planck Institute for Brain Research in Germany. From 1996, he studied the molecular mechanisms underlying motor learning at RIKEN, and in 2001, he joined St. Jude's Children's Hospital, USA, to investigate cerebellar synaptic plasticity. In 2004, he became an associate professor at Kanazawa University, launching research on neurodegenerative disease using lentiviral vectors. Since 2006, he has been a professor and chair at Gunma University, focusing on AAV-based gene therapy for neuropsychiatric disorders. In 2019, he was appointed director of Gunma University's Virus Vector Development Research Center, leading the development of cell type specific AAV vectors in the brain. Dr. Hirai, please.
Okay, I'm grateful to the organizer for the opportunity to share our work with you today. I'm Hirokazu Hirai from Gunma University. Today, I will talk about our efforts to develop AAV vectors that can achieve cell type specific and efficient gene expression in the brain, an essential step for both basic research and gene therapy. This table summarizes promoters and enhancers that can drive gene expression in specific brain cell types using AAV vectors. Those shown in red were developed by our group. Among them, I will focus on two. The L7-6 promoter, which enables highly specific expression in cerebral Purkinje cells, and the mouse EVA1 cassette, which targets microglia. These tools are designed to overcome some major limitations in brain-targeted gene delivery. My motivation for developing a Purkinje cell-specific vector goes back to my early career. I was fortunate to work under Professor Masao Ito at the RIKEN Brain Science Institute from 1996 to 2001. He was a pioneer in cerebral motor learning who not only proposed the theory of long-term depletion, LTD, in Purkinje cells, but also experimentally demonstrated its induction. Back then, I was trying to figure out the molecular mechanisms of LTD. To do that, I needed a way to get a foreign gene into Purkinje cells specifically and efficiently. But at that time, that was technically very difficult to achieve. In 2001, during my time at St. Jude Children's Research Hospital, I encountered the L7 promoter, which was known to drive transgene expression specifically in Purkinje cells. However, it was too large, around 3 kb, and its activity was too weak for AAV-based applications. So we began working on an optimized version. After years of work, we developed the L76 promoter, this one, a truncated 844 base pair version that retains strong Purkinje cell specificity and much higher promoter activity. Using AAV PHPV carrying GFP under the L76 promoter, intravenous injection in mice resulted in selective GFP expression in Purkinje cells. Direct injection into marmoset cerebellum showed the same specificity. This success was featured on the cover of Molecular Therapy, Methods and Clinical Development. To demonstrate functionality, we applied this system to a disease model. In spinocerebellar ataxia, SCA models, expression of the transcription factor, retinoid-related orphan receptor alpha, ROR alpha, is reduced in Purkinje cells. So we use the L76 promoter to express microRNA and to knock down ROR alpha selectively in these cells. As you can see here, Purkinje cells expressing GFP, namely those expressing microRNA targeting ROR alpha, showed either a complete loss or markedly reduced immunoreactivity for ROR alpha. These are the control Purkinje cells showing clear immunoreactivity for ROR alpha. So, as expected, we saw disrupted morphology, misaligned somata, and dendritic atrophy, along with clear motor ataxia in behavioral tests. This video is from control mice, received injection of AAV expressing scrambled microRNA, showing very good performance. So this is mice, affected mice, received injection AAV expressing ROR alpha targeting microRNA. So you see clear motor ataxia. So this shows that L76-driven AAVs can be used not just for labeling, but also for disease modeling and therapeutic gene delivery. Okay, now let's turn to microglia. These immune cells play a central role in brain homeostasis and neuroinflammation. However, because microglia actively eliminate viruses, they've been historically difficult to transduce with viral vectors. After 7 years of trial and error, we finally developed a microglia targeting AAV in 2022, which is published in Communication Biology. We started with a promoter for mouse ionized calcium binding adapter molecule 1, commonly abbreviated as EVA1, which is selectively expressed in microglia in the CNS. However, we found that it also shows some off-target activity in neurons. To suppress this, we incorporated microRNA target sequences, MIR9 and MIR12923P, downstream of WPRE element. These microRNAs are expressed endogenously in neurons, but not in microglia. So upon their binding to the target sites, the viral mRNA is cleaved in neurons but remains intact in microglia. With this design, we achieved microglia-specific expression in the cerebellum and striatum. However, in the cerebral cortex, we still observed substantial off-target GFP expression in neurons, as indicated by white arrows, these white arrows. Our graduate student, Ryo Aoki, tested many variants, but we couldn't eliminate this problem at first. After considering several possibilities, we arrived at the hypothesis illustrated here. If the miRNA target sites are placed after the WPRE, this one, cleavage may still leave the WPRE intact, allowing the mRNA to remain stable and continue producing GFP. In contrast, placing the targets between GFP and WPRE here leads to cleavage in that region. The resulting mRNA lacks both WPRE and the poly-A tail, making it unstable and prone to degradation. This was our strategy to suppress GFP expression, specifically in neurons. So we tested these constructs in vivo. When both WPRE and poly-A were present, this one, GFP expression was very strong. This is a result of using an AAV with MIA-T placed after WPRE, previous research. And WPRE alone could still support expression. But when both WPRE and poly-A were removed, GFP expression was abolished, supporting our hypothesis. Next, we made AAVs where the miRNA target sites were inserted between GFP and WPRE or both sides of WPRE. This simple structure change resulted in dramatic improvement. Neuronal expression was suppressed, and microglia-specific expression was achieved. Immunostaining confirmed that placing target sites on both sides of WPRE eliminated almost all neuronal expression, while boosting both specificity and efficiency in microglia. We achieved over 90% of specificity. This is a graph of specificity and significant improvement over the previous design, this one, and efficiency as well. So we use this AAV system to express GCAMP, a calcium sensor, in cortical microglia. Upon ATP stimulation, intracellular calcium levels rose, visualized in real time. Okay, now ATP is applied, you can see a quick elevation of intracellular calcium level. So this proves the system is functionally compatible with in vivo imaging. Okay, here we express the GFP in microglia, in the motor cortex. After ATP application, you can clearly observe dynamic movement of microglial processes. Here you see. Okay, once more. ATP is now applied. You can see dynamic movement of microglial processes. This provides a powerful tool for lab-cell imaging of microglial behavior. So to summarize, we introduced our developed two AAV tools, the L76 promoter, which allows efficient and specific gene expression in Purkinje cells, and an optimized microglia targeting cassette with miRNA target placement strategies to suppress neuronal expression. Both vectors are or will soon be available via other gene with these numbers. Okay, this is the final slide. I'd like to thank our team, especially Hosoi here, Okada here, Konno, and Aoki for all their great contributions. This work was supported by Brain Minds 2.0, JSPS Kakenhi, and Gunma University's internal research programs. Thank you for your attention. I'd be happy to take any questions. Thank you.
Thank you, Dr. Hirai. Our second speaker, Dr. Karin Kojima, is an associate professor for the Department of Pediatrics at the Jichi Medical University in Japan. She is a pediatrician, pediatric neurologist, and clinical geneticist. She received her PhD in 2014 at the Jichi Medical University. Her key contributions focused on clinical trials of AAV gene therapy for AADC deficiency in Japan, developing AAV-based gene therapy approaches for Rett syndrome and MECP2 duplication syndrome. Dr. Kojima, please.
Good morning, everyone. Thank you very much for the kind introduction, Professor Mizukami, and I'm honored to be invited to speak at this webinar. My name is Karin Kojima, and I'm from Jichi Medical University. Today, I'd like to talk about gene therapies and the development for pediatric neurological diseases. At Jichi Medical University and with many collaborators, we child neurologists are trying to establish new therapy for children's diseases. Gene therapy for AADC deficiency were already in clinical trials. We are preparing for clinical trial for GLUT1 deficiency, Niemann-Pick disease type C, etc. A clinical trial for GLUT1 is scheduled to begin within the next few months. And new treatment for Rett syndrome, MECP2 duplication syndrome, tyrosine hydroxylase deficiency, and centobiban are under development. Today, I'd like to speak about gene therapy for ADC deficiency and NPC mainly. First, ADC deficiency is an autosomal recessive neurotransmitter disorder. In the world, around 200 patients were diagnosed in Japan. 11 patients were diagnosed. In Japan, ADC means aromatic amino acid decarboxylase. ADC is the enzyme to catalyze L-DOPA to dopamine and 5-hydroxytryptophan to serotonin. ADC deficiency presents with disturbed voluntary movements, oculogyric crisis, dystonia, and intellectual disability. Severity are classified as 3 types. 80% are severe phenotype. They are bedridden for whole life. 5% show mild phenotype. They can work independently with dystonia and have mild intellectual disability. Moderate type is between them. This is a summary of the patients who received gene therapy in Japan. 8 patients are severe. 2 patients, patient number 3 and 10, are moderate type. Age at gene therapy are from 4 to 19 years old. Their gene variants are variable. This is a treatment vector. Human ADC gene was inserted into AAV vector. We injected this vector into bilateral putamen by stereotactic neurosurgery via holes of frontal area, 2 points each. Amount of vector was 2 times 10 to the 11th power. This is a post-treatment course of severe type patients. All patients' motor function improved. 75% of patients can walk after gene therapy with support. So their dystonia, so their dystonia has disappeared, and some patients can take oral intake. I showed the movie of severe type girl. Before gene therapy, she was bedridden. She could not move. But 5 months after gene therapy, she started to walk with walker. 6 years after gene therapy, she can walk with assistance. Like that. Next video is moderate type girls. Before gene therapy at 5 years old, she could sit for a few minutes and walk with support. But 1.5 years after, she can ride a bicycle and play a swing. Now she goes to school by herself every day. This is a post-treatment course of moderate type. So, involuntary movements oncologic crisis disappeared in all patients. And one patient got verbal communication skills. This is a short scale of psychological development. We attracted the development content of language sociality function and cognitive adaptation function. DQ 100 is an average of the age, and lower 70 is considered as intellectual disability. Patient 3 DQ increased over 80 into normal range. This is an FMT PET imaging. PET imaging with a specific ADC tracer called FMT revealed that increased uptake persistent for 5 years after the treatment. The putaminal area can be topographically divided into organized subsections connecting to cortical areas. We investigated the association between the motor performance and network connection. The highly transduced putaminal area connection to the prefrontal cortex was associated with improved motor performance at 6 months post-treatment, suggesting that putaminal dopamine promotes the development of an immature motor control system. This is Alberta Infant Motor Scale Ames graph. We evaluated Gold's motor function by using this scale. The Ames scores increased to more than 20 points in patients treated at the younger group, and to more than 10 points in patients treated at an older age. The illustration for ADC mutations of patients. Light blue circle means catalytic residues. Mutations of moderate phenotype are located outside of catalytic residues. It is possible to correlate mutation site and improvement. This is a summary of ADC deficiency. So we performed gene therapy for 10 patients, including 2 patients with moderate type. Motor function improved in all patients during up to 9 years of observations. The level of improvement may relate to age and gene mutations. The next part is gene therapy for NPC. Niemann-Pick disease type C, NPC, is an autosomal recessive lipid storage disorder caused by gene mutations of NPC1 or NPC2. 95% patients have NPC1 mutation. NPC1 and NPC2 are working on cholesterol transport in lysosome. So, this deficiency caused lipid accumulation in tissue, especially brain, liver, spleen. Patients show ataxia, motor delay, psychiatric disorder as neurological symptoms, and also have liver failure, jaundice, and hepatosplenomegaly. NPC patients are clinically divided into 5 groups by the time of onset, from the perinatal group to the adult group. Perinatal and the early infantile group are more severe than older ones. All groups have neurological or psychiatric problems. About prognosis, patients regress after their onset and die in 5 to 10 years. Treatment. Migostat, glycosphingolipid synthetase inhibitor, is an only medicine for NPC, but it can only slow the disease progression a little. There is no definitive treatment, so AAV gene therapy is expected. These are liver pathological slides of our patient. She was perinatal type. At day 59, she received liver transplantation from career father for severe acute liver failure. At 1 year and 6 months, she was diagnosed as NPC and started to take migostat. By Philippine staining, cholesterol accumulation are detected with blue fluorescence. Even in transplanted liver, cholesterol reaccumulation had progressed gradually. She died at 8 years old. I will show the data about gene therapy for NPC. Most of this work was done by Dr. Kurokawa. The vector is modified AAV9-3 vector increased CMV promoter with human NPC cDNA. Vector was injected into left ventricle and cisterna magna at 4th day after birth. We also made AAV9-3 vector carrying on GFP. I show the picture of GFP expression in the brain at 3 weeks after vector injection. GFP was expressed in broad brain area. To compare treatment effects, model mice were divided into 5 groups. Untreated knockout mice could not move toward feeding box because of disease progression. After that, they starved to death. So, girl feeding were put in the cage from 9th week to to prevent starvation for these 4 groups. Treatment vector was injected to wild type and knockout mice. Saline was injected to knockout mice. These 2 groups were not received any injection. The difference between them was with or without gel feeding. First, this graph shows average lifespan of each group. Average lifespan of knockout mice was prolonged 25 days only by gel feeding, and AAV-treated NPC knockout mice have lived 105 days longer on average. Maximum lifespan of AAV-treated knockout mice was 310 days. Next, this graph is body weight changing. At the starting point, there were differences between groups. All 3 untreated knockout mice lost body weight from 5th to 7th week till they died. However, AAV-treated knockout mice showed body weight gain till 15th week and then lost body weight, but mildly. Here is the result of rotor-rod performance. Results of every untreated knockout mice were similar. I chose saline-treated knockout mice group as a control here. Motor function of untreated knockout mice got worse rapidly from 5th week. However, AAV-treated knockout mice maintained their motor function for long. To see the result of each group at 9th week, for an example, difference between untreated and AAV-treated knockout mouse is obvious, and there was non-significance between AAV-treated knockout and wild type. This is movies. So this group untreated knockout mice. Untreated knockout mice showed ataxia at 8 to 9 weeks, and mice could not walk at 12 weeks. But AAV-treated knockout mice could walk smoothly even at 34 weeks. At 42 weeks, neurological symptoms and spasticity progressed, but can walk weakly. I show pathological findings in cerebellum at 11 weeks. In untreated mice cerebellum, most of Purkinje cells disappeared due to degeneration. However, in treated mice, some Purkinje cells were preserved. These are liver pathological staining slides at 11th week. Cholesterol accumulation was seen as blue signals in Philippine staining untreated mice liver. However, in liver of AAV-treated knockout mice, blue signal was reduced. AAV vector was injected to brain only, but also effective for the liver. I will summarize my presentation. Cisterna magna injection can treat neuropathy and liver damage, which are main symptoms of NPC. In preclinical studies, there were no adverse events in rats and monkeys. We will continue to search for biomarkers that can be used as an indication of treatment efficacy. And after vector production, the clinical trials will be started. We are challenging to find treatment of pH deficiency. Thyroxine hydroxylase pH is a very important enzyme to convert tyrosine to L-DOPA. TH deficiency caused due to mutation in TH, so dopamine is not produced. So the patient showed motor dysfunction. We are trying to find gene therapy. And by using AAV vectors expressing TH, ADC, and DCH. And I also trying to develop for Rett syndrome by using AAV vector carrying CRISPR dead-cat system. I appreciate many collaborators and the patients and their family. Thank you for your listening.
Thank you, Dr. Kojima. What a great presentation. Now we're entering the Q&A session, and there's one question to Dr. Hirai. Can you read this? I'll read this. Why was the microglial specificity of the original AAV vector lower in the cerebral cortex?
Okay, yeah, that's a very, very good question. I think there are two main reasons. First, the cell type specificity of the EVA1 promoter varies across brain regions. For example, in the cerebellum, the EVA1 promoter alone achieved 86% microglial specificity, but in the cortex, it was only 2%. Second, the expression levels of MIR9 and MIR129 differ by brain region. As a result, off-target expression was not sufficiently suppressed in the cerebral cortex. That is what we are now thinking.
Thank you. Okay. One more question is to Dr. Kojima. I will read this. Thank you for the interesting presentation. Let me ask a question. How did you inject AAV medicine to patient in clinical test? Direct injection to brain after opening skull or intravenous injection?
Thank you. Thank you so much for great question. So, yeah, I think the question for ADC deficiency gene therapy. So ADC deficiency gene therapy, we performed we injected inject directly to brain, injection to brain, and after opening its skull, but make small hole, so in front front of head, so diameter around 1 centimeter. So and after that, after opening the skull, we inject a special cannula to putamen and inject the cannula, and after that we inject the AAV vector solution. Thanks.
Thank you. Yeah, so that's a stereotactic technique by a special neurosurgeon. Yes.
Do you have any plan to your technique to different disease patients right now, except for AADC deficient patient and Niemann-Pick disease patient, except for those?
Yes, so we are planning to gene therapy by stereotactic neurosurgery for gene therapy for TH thyroxine hydroxylase deficiency. So yeah, it's the same way. So the same method of ADC deficiency.
How many
So
patients are there in Japan or worldwide?
yeah, now, yeah, that's a very rare, super rare
Mhm. Oh,
disease, but 5 patients in Japan I
oh, I
found,
see.
and yeah, in the world, around under 100 patients. Very.
Mm.
Mhm.
Is the disease occurring in the younger age or in adult in TH
Uh,
deficiency?
yeah, so TH
Yeah,
deficiency, yeah, occurred in infancy, yeah,
yeah, yeah.
very young age.
Like ADC.
Mhm.
Okay, thank you. Uh, we have another question. Thank you for asking many questions. And in case of NPC, how long
Mhm.
did the efficacy of gene therapy go on? And redose is needed, or is it possible?
Mm. Thank you so much for your great question. So for NPC, we consider the efficacy of gene therapy continue to for long life. So I think the one-time injection is needed. Only one-time injection is needed. Redose, so we don't think about the redose, and yeah, so because um, so every expression observed so after 15 years over after injection. So yeah, and so we think we don't think about the redose.
In the long-term efficacy, as for the long-term efficacy, yeah, the 15 years has demonstrated. Are there any
Mhm.
decrease in efficacy, or still that's stable?
Yeah, about ADC deficiency. So we checked the FMT PET
Mhm.
analysis. Mm, so yeah, so like over 5 years, and yeah, last one is think yes, 7 years. Yes, it's still expression is still robust after 5 years and 7 years. So yeah, I think the efficacy is still go on. Maybe
So you hope it will
yeah. Okay,
stay
I know.
stable and
Mhm.
forever, as long as the life.
Yes.
Yeah. Okay. Thank you. Any questions? Mm, In as for the redosing, do you think theoretically is it theoretically possible to redose in the brain, even in the presence of neutralizing antibody produced after the first injection?
Mm. Yeah, so it's a very important thing. So, but if the if the if we use another vector, so another serotype vector, so for redose therapy, so yeah, at that time, so we can do redosing, so carrying on same gene, but and other, so yeah, so another serotype of AV AV vector with carrying same gene. Yeah, it is if it is possible to affect.
Thank you. Hmm. Hmm.
As for the neutralizing antibodies, I read a recent paper. Actually, uh, my lab members introduced one paper. So, high dose injection of AAV vector causes B cell infiltration into the brain and produces antibody. So, but changing serotype capsid is very effective to evade uh, evade neutralizing. So Dr. Kojima's opinion is very, I think, correct.
Thank you so much.
Yeah, yeah, I think so.
Yeah, I agree. Thank you. Uh, let's move on to Dr. Hirai's work discussion. And you have tried the promoters. You have the promoter specificity, and you also may try to shorten the promoters and to still to improve the efficacy by the promoters. Uh, what do you think about the results about your research to succeed in the promoter specificity by the promoter?
Sometimes goes well, but sometimes my strategy didn't work so well. We usually start by identifying a gene that's selectively expressed in the target cell type, like EVA1 for microglia. Then we clone up to 3 kb upstream of the start codon as a promoter candidate. Since 3 kb is close to the AAV packaging limit, actually AAV packaging limit is 4.7 kb
Yes.
or around 5 kb.
Yeah.
But we have to consider space for WPRE, poly-A, and gene of interest, and ITR. So removing those spaces, remaining space is around 3 kb. So 3 kb is the close to the AAV packaging, actual AAV packaging limit for promoter candidate. And we gradually shorten it based on transcription factor binding sites, and test each version by making AVGFP and injecting into mice for in vivo screening. But sometimes goes very well, but sometimes doesn't work so much. So in that case, we have to change genes but completely, or we have to use microRNA target strategy. But okay, yeah, that's what
Yeah,
we are
thank you
doing.
very much. Yeah. My question was not very focused, but I have one more question concerning the similar issue. How can you evaluate the difference of the capsid in transducing the specificity of the brain cells?
So you mean
Neurons.
importance of capsid?
Yeah. Yes. The capsid. Can you create any specificity by changing the capsid?
Yes. Yeah, that's a very important point. If the promoter is highly specific, the capsid doesn't matter so much. But if the promoter lacks strong specificity for the target cell type, so in such case, we have to use capsid with high tropism for target cell type. So many cell type specific promoter do not have so much specificity for target cells. So in such case, we have to carefully choose capsid, which has high promoters, high tropism for target cells.
Can you actually name some of the good examples of the specificity of the capsid to a certain cell type?
Okay. Yeah, it depends on a species, like mice or marmoset. But in the case of marmoset, I think, for example, MVP promoter for oligodendrocytes, myelin-based protein promoter for oligodendrocyte. AAV, I think AAV5 is best. So if you use AAV9 or different serotypes, specificity drops significantly. So selection of capsid serotype capsid is critical, I think.
Thank you. Thank you. Yeah, we have a couple of questions coming from the web, and the similar questions. Professor Hirai, did the microRNA affect the overall expression levels of the transgene?
Mm, yeah, yes, I think so. But I cannot completely understand the point of this question. But microRNA, microRNA is, in my case, in microglia's case, MIA9 and MIA129 expressed with high levels in neurons. Okay, and then viral mRNA with target sequence is degraded. So expression of transgene is suppressed drastically. But microglia do not have MIA9 and MIA129. So viral mRNA is intact, even to expression of transgene. So, for example, oligodendrocytes, astrocytes, other serotypes, if MIA9 and MIA129 are expressed, expression levels are suppressed. But yeah, that is. I'm not sure whether my answer is. I'm answering the question, but
Thank you. Yeah.
Yeah.
Thank you very much. Let's move on to the next question to Dr. Kojima. In NPC case from the Rotolod test, AB-treated group showed marked efficacy, but decreased gradually. Can the results support one-time administration in clinical setting?
Oh, thank you for great point and great question. But yeah, yeah. So yeah, it is possible to need to redose so more administration, yeah, in clinical case, but we don't have the data about two times administration. So but yeah, it is possible. It is possible to need so redose, but yeah, but now. So yeah, our data, our lot of data show the so efficacy, but yeah, so sometimes after and yes, so compared to wild type, the knockout mice motor function decreased gradually. Yeah, but yeah, so yeah, it is so tough question. Yeah, but yeah,
Yeah.
so we but yeah, we have to monitor so so long time for the knockout mice experience, and after that we can answer this question. Sorry.
Yeah.
Mm.
Thank you. In the NPC case, mice, what was the dose? And did you performed the, yeah, in the earlier experiments, maybe you tried to determine the optimum dose. Yeah. Do you think the higher dose may be more effective?
Yeah, maybe. I think, yeah. So if the clinical trial, if when we start the clinical trials, we are planning to dose setting, so high dose and low dose. So
Mhm.
yeah, so maybe the if there is no side effect, the higher dose is better. I think so.
Mm. Yeah. Okay. Uh, are there any questions from the audience or each other? Okay, I may make one more question concerning the ADC. And
Mhm.
you mentioned that the ADC is very rare, and uh, worldwide in the 200 patients so far found. Uh, are there any geographic specificity in the location of the patients?
Yes, thank you so much for your question. So, yeah, so geographic specificity there is. So, in Taiwan, and so the um, third or yeah, one third of patients lived in Taiwan. So, because so they have same splice variant and of the ADC gene. So yeah, and so the 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 splice variant gene cannot produce any ADC enzyme. So they are in Taiwanese patients very severe type. Mhm.
So, except the Taiwanese case,
Mhm.
all of the cases are sporadic. The majority of the
Yes.
cases are sporadic mutant, mutant.
Yeah. So there are many variants were reported. Mm.
Mhm. So, in Japan, there are 11 patients so far found. Yeah.
Yeah.
Uh, so, you treated most of the patients, is that right?
Yeah, but now, so we we don't find we didn't find other patients in Japan.
Oh,
So, oh, but in the future, so we find the new patient I want to treat. Yeah.
okay. Thank you. Uh, any more questions so far? No questions from the chat Q&A. And if we have the questions between us, yeah. If not, yeah, I think it's time, almost time. So I will try to close this session. Thank you for attending today's webinar. Thank you to our speakers for sharing your expertise and for a great discussion. This event was recorded, and you'll receive an email with a link to watch the recording when it's available. I would also like to invite everyone to join ASGCT in their upcoming molecular therapy webinar series, which will be taking place virtually on April 23rd. Finally, ASGCT values your feedback and welcomes suggestions for our future sessions. Please take a moment to complete the survey linked on the right of the screen under evaluation. Thank you so much.